Jai Sri Ram, Jai Sri Ram, Jai Jai Sri Ram. Hello everyone, my name is Gyanendra Kumar. I am biology educator in India. And few days I was really upset. My friend Deepak Singh, uh, he was in Indian Army. He was a national security guard also. Um, he lost his life. He sacrificed his life in Jammu and Kashmir. I was very upset. That's why I'm not posting video. I'm sorry for that. Now we'll continue it. So in this video we will see structure of human eye. This human eye, this human eye, we all know that this human eye present in our skull, isn't it? In skull there is one socket. That socket is called eye socket. You call it eye socket, or you will call it orbitolong socket. Okay, in this orbitolong socket only human eye present. Fine. And this human eye is for visualization, and it is moved by six muscles: superior oblique muscle, inferior oblique muscle, superior rectus muscle, inferior rectus muscle, and then external rectus muscle, and then internal rectus muscle. This six muscle, control movement in eye, and they control by three cranial nerves. We all know we have 12 pair of cranial nerve. Cranial nerve third, and then we have cranial nerve 4, and then abducens nerve we have sixth cranial nerve. These three nerve, they control this all skeletal muscle, and they finally control movement in eye. Fine. Now you come back to the eye. This eye, okay, fine, this oval structure, and this our eye have three layer. Outermost layer of the eye is called fibrous tunic. We are calling it fibrous tunic. Okay, and then next layer, that is called uh, vascular tunic. Inner layer, next layer, outermost layer is fibrous tunic. Then next layer, which is inner layer, is called vascular tunic. And then innermost layer we have, that is called retina. Okay, so we will see these all layers one by one. Let's start with the outermost layer. That outermost layer is actually called vascular tunic. Sorry, fibrous tunic. This fibrous tunic, it have two parts. Our fibrous tunic have two part. What are that part, students? That part is one part, which is inner part. We will call it sclera. Okay, this inner 5/6 part of the fibrous tunic is called sclera. And then they have outer 1/6 part also. That outer 1/6 part is called cornea. Okay, so this sclera is mesodermal. You see, the sclera is mesodermal origin, except sclera and uvea. What is uvea? We'll see. Except sclera and uvea, remaining everything is ectodermal origin in our eye. Fine. So our sclera is actually mesodermal origin, and it is white color layer. Okay, and it's tough layer, So connective tissue layer. Why? Because it's rich in collagen fiber. Because of presence of collagen fibers, okay, this white color fibers is they are going to protect our eye. Fine. Now the same sclera when the same sclera go to anterior part of the eye, they modify and they form one transparent structure. This transparent structure is called cornea. Okay, this cornea is for refraction of light. Yeah, it is for cornea is for refraction of light. And one unique thing about cornea is what, the cornea transplantation is highly successful. Reason is what? The cornea is non-muscular layer. It is very easy to transplant them. There will be no chance of organ rejection here. No organ rejection here. Organ rejection here, right? We all know that organ, or what is organ rejection. When organ transplant to the host body, the host body T lymphocytes, they attack on that organ. They consider it as a foreign invader, and who attack? T lymphocytes attack. Okay, T lymphocytes attack. We will see in immunity in detail. Fine. So this cornea have no blood supply, and when it have no blood supply, no question of no, our T lymphocyte will not reach there. No question of the the rejection. Fine. So this is what our outermost layer, that is fibrous tuning. The next layer, which is vascular coat, we are calling is vascular tuning. Either you call it vascular tuning or you call it uveal tract. Fine. This uveal tract is deeply pigmented layer. It's highly pigmented layer. Okay, rich in melanin pigment, and it reduces unnecessary refraction of light inside our eye. Yeah, unnecessary, because see, when the light wave entered through our eyes, they can reflect in many direction, here there, unnecessary. So they are going to prevent that, because they are hyper pigmented, so they can do it. Okay, now this our vascular tuning have two part. The part which is present inside, okay, inner 2 by third part is called choroid. We are calling it choroid. And then same choroid outside they modify, and anteriorly they modify, and they form one structure that is called iris. Fine. So choroid, basically what they do, they are hyperpigmented, they prevent refraction of light. Okay. Now this same choroid, as we discussed, 1 by third part of the choroid, our vascular tuning, they modify and they form iris. What is iris, students? This iris, you see your eyes in mirror. In this eye, in center, one black color part present. That black color part is only called iris. Fine. And obviously this iris have a particular color. This iris color, is controlled by genetics of the person. Okay, is pigmented, it have a particular color, and that color of the iris is controlled by genetics of person. Different gene, it's not one gene actually, multiple genes involved in that. Someone has brown color eyes, someone have black color eyes, someone have so you say that someone, oh my god, he have blue color eyes. So it's controlled by different gene. As for example, in the 15th chromosome of the person, yeah, in the 15th chromosome of the person, there is one gene. This gene is called OCA2 gene. This OCA2 gene, they form one protein. That protein is called P protein. Yes, P protein. This P protein help in, it's actually involved in melanosome maturation, and they only decide amount and quality of the melanin pigment stored in that region. Fine. OCA2 gene present on the 15th chromosome. OCA2 gene present on the 15th chromosome, and they form P protein, and this P protein involved in the melanosome maturation, and amount and quality of the melanin should stored there, that also decided by them. So this gene have a crucial role uh, in deciding color of the eye. And then we have one more gene. That gene is HERC2 gene. Okay, this HERC2 gene have also, it's control the expression of the OCA2 gene. Actually nearby 150 gene control the iris color. Fine. Okay, that's why we are saying that. Okay, fine, it's example of multi-gene inheritance. But these are the two crucial gene, OCA2 gene and HERC2 gene actually control expression of the OCA2 gene. And finally they decide the color of eye. Fine. Are you observing in center of iris one black color dot present? This black color dot, is called pupil. Fine. We are calling it pupil. This pupil, this pupil decide amount of light received into the eye. Fine. This pupil actually, this is our iris. In center of that iris we have another small dot structure, black color structure. This is actually pupil. This pupil will decide what amount of light will go to the retina. Fine. If the light is too bright, okay, so pupils become smaller, okay, and when light is dim our pupil will dilate, isn't it or not? And they are control this dilation in this, so this dilation, is controlled by different muscle. Okay, we will see when time will come, we'll see that on detail. Fine. Then this same vascular cord, they modify and they are forming one structure that is called ciliary body. Either you call it ciliary body or you call it ciliary muscles, right? So just behind the peripheral margin of iris, they are modifying and they are forming one structure called ciliary body, you say, or ciliary muscle, you say. The ciliary body or ciliary muscle, they actually the control the thickness of our lens. Okay, they have some ligament, that ligament is called suspensory ligament. So by the help of suspensory ligament, they control actually thickness of the lens. You see here in this video, you see, one second, you see. Okay, see, this is our the ciliary muscle. Fine. The ciliary muscle, and this ciliary muscle is modification of the our vascular tunic. And then they have, this is suspensory ligament. By the help of suspensory ligament, they control the thickness of our lens. whenever suspensory ligament Whenever suspensory ligament contract, our ciliary muscle relaxes, and our lens, it will get more and more stretched. Yes, lens will get stretched, and it will become, it will become thinner. Yes, the lens become thinner. Okay, it will become more flat. Whenever suspensory ligament relaxes, okay, our ciliary muscle will contract, and when ciliary muscle will contract, then lens will become more globular, it's become more thicker. This is how it will work. So they are going to, it is the vascular tunic ciliary body who control the thickness of our human eye lens. Fine. If we'll summarize total that all information till now we have studied, so our eye have three parts, three layers: fibrous tunic, vascular tunic, and then innermost layer, retina, that we will see in detail. This fibrous tunic, which outer layer is avascular layer, avascular, and it's the dense, is rich in the collagen fibers, dense for white color fibers. And it have two part. First part is sclera, that is inner posterior 5 by 6 part, and then we have cornea, anterior 1 by 6 part. And then you come to vascular tuning. Vascular tunic is highly vascularized and hyperpigmented region. Okay, and it's originated from mesoderm. And they have total three part. First part is called choroid. Choroid is present inside, we are calling it uvea, right? And this uvea is hyperpigmented layer, it's prevent refraction of light. And then we have ciliary body. The ciliary body, is this control the thickness of our eye lens. And then we have iris. This iris, this iris is behind the cornea actually. And this iris is decide color of our eye. And this iris have pupil. The pupil dilate when light is dim, and the pupil constrict when light is bright. Okay, and then we have innermost layer, that is nervosa coat, and we will call it retina. Fine, we'll call it retina. Now come to retina. This retina have three part. First part is optic part, second part ciliary part, and third part, iridial part. This optic part is very important. This optic part, actually is a pigmented part. It have some pigmented structure. Fine. Actually it has cells, and that cells are rich in melanin pigment. So you may ask, sir, vascular tunic have also melanin pigment, and here also melanin pigment? Yes, both part have melanin pigment. And next part is normal part. This is mainly for visualization. Why? Because this part have, in this nervous part we have rod cells, and then we have cone cell. This rod cells and cone cells have ability to detect the light. Fine. So as they have ability to detect the light, they can absorb the light, so they are only for responsible. Fine. See, this portion, let me show, this portion, is nervous part. This portion is nervous part. Why it's nervous part of retina? Because it have rod and cone cells. Fine. And then this portion, you see, this portion is pigmented part. Pigmented part means it do not have rod and cone cells. It have actually, it have also actually melanocytes, and that melanocytes are rich in melanin pigment. And this is the point of transition of this nervous part and pigmented part. I'm writing here, this is pigmented part of the retina, rich in melanin melanocytes. Okay. And this is nervous part of the retina, nervous part of the retina. Fine. This pigmented part, students, this pigmented part, obviously it have also role, and the point of transition of nervous and pigmented part is called aura serrata. Aura serrata we are calling it. Fine. Now we see between the, this is our lens of the eye, and this lens is convex lens, and it's made by crystalline proteins. Yeah, it's made by crystalline proteins. Okay. It's transparent structure. Okay. It's a transparent structure made by crystalline protein, and we discussed this thickness. Their thickness is controlled by, whenever required, they can be globular, they can more flat, and if they are controlled, thickness is controlled by our ciliary muscles. Now you see, there is one chamber between this our lens and retina. This chamber is called vitreous chamber. This vitreous chamber have a jelly like material that is called vitreous humor. Yes, they have a jelly like material that is called vitreous humor. This vitreous humor, is rich in hyaluronic acid, and it form only once in life, right? And it's form only in embryonic stage. Once it was formed in embryonic stage, it will never form again. Fine. And because of this jelly kind of material only, our eye have a particular shape. Okay. So this vitreous humor which is present, vitreous chamber have vitreous humor, vitreous, and that vitreous humor decide, a particular shape of the eye. Then you see, this is cornea of the eye. Okay. Between cornea and lens there is another chamber. This chamber is called aqueous chamber. Okay. Or we are calling it anterior chamber. This aqueous chamber have a fluid that is called aqueous humor. Aqueous humor is such a kind of fluid, in whole life it will form. Okay. It will form, it will exit, it will form, it will exit, and it will exit through fine canals. Okay. And this canal is called canal of Schlem. Schlem is, it will exit, it will form, when it will get older it will release out through this canal, and this canal is called canal of Schlem. So in this video we discuss structure of the human eye, yet it is not completed. Remaining we'll see in next video. We will see retina, then we will see mechanism of vision in next video. Meanwhile, you focus on your study, learn everything, subscribe my channel. Bye.